Good morning, everyone. Welcome. Thank you for joining us at today's webinar. We are going to get started just past the top of the hour. This is a Zoom webinar, which means that we can't see or hear you out in the audience. If you have any questions for our panelists today, please use the Zoom Q&A function or the YouTube chat in order to ask those questions. And then our speakers are going to address them. They'll either type an answer to you during the other talks, or they will answer it during the panel session at the end. So we're going to get started in just a moment at the top of the hour. Thank you for joining us here on Zoom or YouTube. And thank you to those of you who are watching the recording of this after the fact on YouTube. We are going to get started in just a couple of minutes. If you have any questions during today's event, please ask those using the Zoom Q&A or the YouTube live chat. So it's now 10 o'clock on the dot here in Pacific time. So I'm going to hand it over to Mike to get us started. Mike?
No, thank you very much, Caitlin. Welcome everybody to Revealing the Mammalian Cortex, our flagship release webinar in which we'll cover all the highlights and further interesting content of this landmark publication. But first, it's my great pleasure to introduce John J. Nye from the NIH, Director of the U.S. Brain Initiative, formerly a professor at the University of California, Berkeley for over 25 years. John played an integral role in the papers prior to kind of accepting his role as director of the Brain Initiative. And John, would you give us some remarks here about your perspective?
Thanks, Mike, for that kind introduction, and welcome everybody to this webinar. I'm really delighted to be here. I'm really delighted at the huge progress that's been made by the field as embodied by these papers over the last 5+ years, using and developing some really bleeding edge techniques to create comprehensive and integrated atlases and cell census of mammalian brains. And what you hear about today is an incredible effort that was stood up by a large consortium of investigators from over 250 investigators from over 45 institutions across 3 continents. And to really understand what is it that makes up mammalian brains at the cellular level and what their properties are, not just one property at a time, but integrating across multiple features, multiple modalities to really give us the resources we need to understand how neural circuits function, how they are affected in disease, and ultimately how we can treat the most devastating human brain disorders with this knowledge that will give us information not just about what's going wrong, but also some molecular insights that can be leveraged for precision targeting. And that is really the long-term goal. You're going to hear about some amazing science, just a few snippets here, but that's been published across over 24 papers in Nature and Nature sister journals. And again, this will give us foundational knowledge, not only for understanding how neural circuits function and brains function, but also for future cures. And much of this was made possible by this incredible team effort. And team science is, at this scale, is really quite new to the neuroscience field. Of course, it's quite common and very, very powerful in other fields such as high energy physics and astronomy. And it's not to say this is the only way to do science, far from it. We still rely on people asking great questions that drive the innovations here, but certain resources really do require people working from across disciplines, across different backgrounds, with different know-how, access to different reagents and resources to really make a project like this happen. And I think you'll see over the next hour, hour and a half, what has really come to bear because of not just having a lot of people work on this project, but people learning how to work together toward a common goal. So we not only gained a lot of knowledge, we also gained knowledge about how to do science. And together, the knowledge resources that we have, the working resources that we have, the ability to work together in these large teams really do set the stage for standing up a human brain cell atlas, which will be coming over the next few years. So without further ado, I just want to turn it back over to Mike and sit back and enjoy some really great science over the next hour and a half.
Thanks, John. Let's advance here. We think have we covered, Caitlin? Do we need any more logistics here, or are we all set?
I think we're good here.
Okay, let's move on then, I guess. Yeah. So there's a big lineup here of speakers here. These are some of the several of the primary authors on these papers and their teams. And I won't go into you'll hear we'll briefly kind of mention the person as they step up. But so to give them the most amount of time, we won't kind of read through all this. Next slide, if you would. This is a picture of the cover here, published online on October 6th and in print on October 7th, 2021. This cover, as in many papers, one goes back and forth with possible cover designs, and we finally settled on this one here, which was we're quite pleased with, and everyone was very supportive of. This, you can go find these papers; they're all available. It's a very comprehensive kind of issue. Next slide. I just kind of summarize in one more slide here, kind of summarizing, I think, that helps just set the landscape a little bit. On the left, as in most kind of big Nature articles, there's a kind of great care taken by the journal to present the materials and popularize it. One of the forms this takes, of course, is the famous news and views, for which outside people not involved in the study are solicited to sort of write about it. And this was a nice article here. We got a very nice news and views from the two authors here. There's sort of a diagram showing it. On the right, this was our extended sort of data figure 1, which basically just describes the overall project kind of architecture, showing the profiling in 3 species, the deepest dive in the mouse, extremely comprehensive, and then sort of some less so in marmoset and human, but with plenty of cross-species deep corroboration in the motor cortex of all 3 species. A variety of techniques brought to bear on this, from deep RNA-seq profiling, MERFISH, PAP-seq, retroseq, whole brain morphology, viral tracing, etc. And you'll hear about a number of these kind of techniques and technologies today. Then we brought this forward, lots of integrative data analysis, and a whole set of resources available, which are available through BICCN.org, our Brain Initiative Cell Census Network resource site. And this contains a sort of a data catalog, allowing you to kind of access all the different teams' data by project, team descriptions, a detailed presentation of the science, of a good deal of the science, which you'll hear about today, is described, referenced in that on that site, as well as access to many of the analysis and tools through our partners, which helps to gain access to the data from the archives and where the primary data lives, and to really make more use of it yourself in your own work. So I think that's all I'll say, and I'll hand it over to our first speaker. So we'll just introduce the speakers as they come. If you advance the slide yet. First, we have Honghui Zheng, director of the Allen Institute for Brain Science, who will speak about a characterization of cell types in mouse primary motor cortex. Honghui.
Thanks, Mike. Yeah, can you advance to the next slide? Yeah, I will give you a very brief overview of the multimodal characterization of cell types in the mouse primary motor cortex. The characterization in the mouse primary cortex is really the starting point of the project, and we generate the atlas as a reference for cross-modality comparisons as well as cross-species comparisons. So here I'll give you a couple of highlights of the different types of data that have been generated to come up with this reference atlas. In the next couple of talks, you'll hear more characterization of cell types using other major approaches. So we began with a single-cell transcriptomic characterization of cell types. In this region, we used a number of different methods, including single-cell RNA-seq and single-nucleus RNA-seq, and using 2 different approaches, the 10x genomics approach as well as the SMART-seq approach. So we come up with this consensus transcriptomic taxonomy shown at the top of this slide, that it is really the anchor point to link all the other kinds of cellular properties to the cell type taxonomy itself. So in the third row there, using the single nucleus RNA-seq data generated from Evan McCaskill's lab at Broad Institute, we are also able to obtain the relative proportions of the different cell types from the primary motor cortex. And furthermore, using the patch-seq method that is done in Andreas Tolli's lab at Baylor, we are able to obtain the local morphologies representative of each cell type, as well as their morphoelectrical properties that can link the molecular identities that identified by single-cell RNA-seq with the literature information of the previously known cell types that have come from many years of research in the field. Next slide. Here, I just want to highlight one type of integrated study that we have done, and this is really combining single-cell transcriptomic and single-cell epigenomic data together to reveal cell type-specific gene regulatory landscape. In this particular study is a collaboration with several researchers at the Salk Institute and the UCSD, as well as the Broad Institute. The upper left corner, we generated a computationally derived integrated taxonomy combining 9 different data sets together, 8 of which are shown here. And those 8 data sets include 6 single-cell RNA-seq data set and 2 epigenomic data set, one for ATAC-seq and the other is methylation-seq. As you can see in the middle panel, through this kind of computational integration, each individual data set is represented well in the integrated taxonomy, so that we can really now integrate all the specific gene expression and chromatin landscape information together. And using one gene as an example to show in the right panel, the TSH2 gene, we can see that the genomic tracks, the genomic signatures for both RNA expression as shown at the bottom, and for chromatin accessibility peaks shown in the middle panel, and as well as the methylation peaks can all be aligned together. And through this, we can correlate gene expression with gene regulatory element, and importantly, then we can discover cell type specific gene regulatory elements or enhancer elements that then can be used to drive generation of cell type targeting tools. Next slide. In a very different types of study, the consortium also came together to derive a input-output wiring diagram of the mouse primary motor cortex at the cell subclass level by combining several different cell type or cell class-based technologies. And this include AAV-based anterograde tracing, rabies virus-based retrograde tracing, as well as axon projection patterns at a single cell level through brain-wide single neuron fold morphology reconstruction. And this highly scalable bar-seq approach that links projection patterns with molecular identities of the injected cells. All this data, very different types of data, are co-registered into the common coordinate framework that again allows correlation and integration of the different data types together. And finally, next slide. And here is another large-scale study specifically focusing on looking at morphological diversity, actually in many different brain regions beyond just the primary motor cortex, looking at the correspondence of morphological and projectional properties with the cell type identities. And we found through the study of 11 major projection neuron types that there are highly distinct projection patterns between those major molecular types. But in addition to that, we also found extensive additional projectional diversity even within each of those molecular types. And that kind of diversity and specificity is reflected at 3 levels, including regional specificity, topographic specificity, as well as variability at each individual cell level. Okay, with that, I conclude my overview of the cell type characterization in the primary mouse primary motor cortex, and I'll pass on to the next speaker.
Well, thank you, Longhui. Next, please. So our team really studies the demethylation, a major epigenetic marker in different brain cell types and also neurons in different brain circuitry. So we have two focuses of our study. One is to use demethylation, single cell demethylome sequencing to characterize different brain cell types and the regulatory DNA landscape within those cell types. Second, we aim to understand the epigenomic diversity of different neurons and connect that with their connectivity. Next, please. So using single cell demethylome sequencing, we can study the genome-wide demethylation status to measure the epigenomic activity, not only in genes, but also in the vast non-coding genomic regions, including regulatory DNA sequences such as enhancers, as Honghui mentioned. So in this study, we analyzed 110,000 cells from 45 brain regions in the mouse, and we used a three-level iterative clustering analysis and annotated at the end 161 different subclusters from the mouse cortex, olfactory area, hippocampus, striatum, and pallidum. Next, please. So in addition to this comprehensive demethylation cell atlas, to further understand the epigenomic diversity of neurons with different connectivity, we developed the method called EpiRetroseq. So EpiRetroseq combines retrograde tracing with single cell demethylome sequencing. In each EpiRetroseq study, the neurons that project to the injection site of the retrograde tracer are labeled with GFP in their nuclei. We then dissect out different regions in the brain that harbor those labeled cells and isolate GFP positive nuclei with flow cytometry and analyze them with single cell demethylome sequencing. Therefore, in the data set for each neuron, we know their cell location from the dissection. We know its external projection from the retrograde tracing, and we also know the demethylome of each individual neuron. Next, please. So using this method, overall, we analyzed 12,000 cortical projection neurons from the mouse brain from 63 different projections to subcortical and the cortical targets. Overall, using all the informations together, in addition to annotate the major cell types in our data, we can start to analyze within each cell type, quantify the differences in methylation between neurons that located in different brain regions, and also quantify the differences between neurons from different projections. Next, please. Here I'm just showing you one example, the layer 5 ET neurons, which are neurons in layer 5 projecting to extratelencephalic targets. So as you can see here in the middle panel, the layer 5 ET neurons now labeled by where they locate in the cortical areas in different colors. So clearly neurons located in different cortical regions, they separate from each other in the demethylation space. In addition, next please. If we focus on primary motor cortex, the MOP neurons here, and we analyze their different projection targets or different ET projection targets, now we can start to really dissect within the same cell type, layer 5 ET, and within the same brain region, MOP, how the neurons with different projection targets may vary in their methylome from this projection. So on the bottom, you can see looking at 5 different ET targets, MOP neurons projecting to medulla clearly have very distinct methylation profiles compared to the other ET projections. And those medulla projecting neurons are enriched in one specific subcluster of MOP layer 5 ET. Next, please. So at last, I want to just emphasize again that from our demethylation studies, not only we can study the epigenetic state of genes, but we also have information of the epigenetic activity of the non-coding regulatory DNA sequences. For example, for each of the MOP layer 5 ET subcluster, we identified a set of genomic regions that have unique depletion of the methylation specific to that particular subcluster. We can further infer which transcription factor might be binding to those demethylation, hypomethylated regions, that those other transcription factors might be really carrying an important role for cell type specific gene regulation. So if you're interested, we have more details in the companion papers and also the flagship paper. Thank you.
Hello. Can I be heard?
Yes.
Yep. Yeah, we have Zhao Wang here from Harvard.
All right. Okay, so you just heard from Honghui and Juju about using a powerful single-cell sequencing-based method to identify cell types in the primary motor cortex and other brain regions. As we know, these cell types are not randomly distributed in the brain, and their spatial organization is important for brain functions. So to map the spatial organization of cell types in the mouse primary motor cortex and its adjacent areas, we used a spatially resolved single-cell genomics approach called MERFISH, developed in my lab. So next slide, please. Yeah, so due to the time limit, I won't have time to describe how MERFISH works, but except to say that MERFISH allows the simultaneous imaging of thousands of genes in individual cells. And then this in situ single cell gene expression profiling capability allows us to identify cell types and map their spatial organization in intact tissues. Next slide, please. And applying MERFISH to the primary motor cortex and its adjacent areas, we identified 95 cell clusters that belong to 23 major subclasses of cells. And these cells adopt complex spatial organization with most distinct cell clusters also assuming distinct spatial distributions. Next slide, please. Yeah, I won't have time to describe all aspects of the spatial organization of cells in the primary motor cortex. So I'll just highlight a couple of example findings. For example, we observed that not only excitatory neurons, but also inhibitory neurons adopt layered organization with many inhibitory neuronal clusters primarily occupying just one or two cortical layers. And next clip, please. And we also observed that intratelencephatic neurons, which is a major branch of neurons, which constitute about 70% of excitatory neurons in the motor cortex, forms a molecular and spatial gradient that spans nearly the entire cortical depths, in which the IT neurons exhibit more or less continuous change of gene expression profile that are correlated with their cortical depth position. So next slide, please. Now, because the MERFISH is an imaging-based approach, we can combine that with other imaging-based approaches to perform multimodal analysis of cells. For example, here we combine MERFISH with retrograde labeling to identify the projection target of molecularly defined cells. As a proof of principle study, we injected retrograde labels into 3 cortical regions, secondary motor cortex, primary somatosensory cortex, and TA region. And then these labels propagate to the primary motor cortex region through the axons. And then in the primary motor cortex region, we imaged not only the MERFISH gene panel, but also the retrograde label in individual cells so that we can identify their cell type information and projection target. Next slide, please. Next click. So with that, we observed that the cortical projections of MOP neurons form this multiple cell type to multiple target kind of a complex network in which individual clusters tend to project to multiple targets, and each target tend to receive input from many clusters. And we hope that this MERFISH approach can also be combined with other tracing approaches to identify connectivity between molecularly defined cell types. And we previously shown that MERFISH can be combined with activity marker imaging to reveal the functional role of molecularly defined cell types. So this type of multimodal analysis, we hope can help us advance the understanding of how molecularly defined cell types form neural circuits to perform specific brain functions. Next slide, please. Yeah, so with that, I just want to briefly acknowledge the people who are involved in this work. And in particular, I want to give a shout out to a very talented postdoc, Meng Zhang, in the lab, who has done most of the experiments and an analysis. And the work is done in collaboration with Hongwei Dong's lab at UCLA and USC, and Hongwei Zhen's team at Allen Institute. Thank you.
Hi, can you hear me?
Yes.
Great. Thank you very much for the opportunity to speak here today. I'd like to describe 2 additional studies within this package of studies that are starting to tackle comparative analyses and human analyses using the same types of approaches that you've heard for in the mouse so far. Next slide. And I'd like to start off by saying that both of these efforts that I'll describe were really big team collaborative efforts across many labs that of the sort that really could only have been done within the BICCN at the moment. So as you heard from Honghui Zhang at the beginning, the single cell genomic technologies, transcriptomics and epigenomics, have been exceptionally successful at being able to create a molecular classification of cell types. And what we were able to show in this study is that the same result can be achieved in frozen archived materials from monkey and from human. And applied to each of these species, we're able to achieve a classification of cell types, which is very similar in complexity across the species. And these data can be quantitatively aligned with one another to create a consensus cell type taxonomy. And there really are, I think, a number of significant results from this. The first, of course, is that it can be done, that this can be taken up to human and more similar non-human primate model organisms, but also that the overall architecture of cell types turns out to be very similar across the species. There really are no major branches of cell types that are distinctive between the species. And this allows us to create this consensus classification where we can compare similar types and align them to one another. And that in turn allows us to predict lots of things that are difficult to measure in human or non-human primate, such as developmental origins of cells or long-range projection neurons. We now have a way to align these and to make these inferences about what kinds of cells are there, and to show that this is really an overall very similar degree of complexity. If you could show the next slide, please. In contrast, despite this overall homology, there are lots of species specializations. And one of the most obvious of these is at the level of the gene expression that we use to define the cells in the first place. So although we're able to very nicely align cell types between the species, many of the markers that actually define those cell types, the most selective cell type genes, are not the same across species. And so while there are a sort of core conserved set of genes that are markers in all species and allow you to do this alignment, it actually turns out that the majority of genes that are highly cell type selective for any given cell type actually vary between the species. So this is really sort of an interesting juxtaposition of an overall homology, but lots of species differences that turns out to really be sort of the general pattern of what we see. That there's this overall homology. You can think of an analogy of a 4-limb homology across species, and yet the 4-limb of a bat and the 4-limb of a mouse or a human has a very different function. And that's sort of the type of thing that we see. So if you could move to the next slide. The other study I'd like to briefly describe tries to move beyond this molecular classification and to look at the properties of cells. And Honghui already described this method of patch-seq applied to the mouse by Andreas Tullius's group. We have taken a consortium approach to trying to achieve the same thing in human tissues, using neurosurgically resected tissues from either epilepsy or tumor removal surgeries that provide live tissues from which one can do slice physiology types of experiments. And the modern version of this is to extract the nucleus at the end of the experiment so that you can sequence that nucleus and pin a molecular identity on the cell. So this so-called triple modality patch-seq method is very effective at being able to characterize these molecularly defined cell types. And as I'll show you here, is possible to do in a human and certainly the non-human primate as well. Next slide, please. So we applied this technique to revisit a very long-standing observation, which is that in human brain evolution, the cortex has disproportionately expanded. And furthermore, the upper layers of the cortex have disproportionately expanded within any part of the cortex. And if you simply look by histology, it's been very clear that, that the diversity of cell types just seen by cellular anatomy is quite more pronounced in human or non-human primate. And it turns out that from the transcriptomics, we also see greater diversity within this population of neurons in the upper layers of the cortex that are known to make connections with other parts of the cortex compared to a mouse. So we applied this patch-seq approach to try to explore this and see whether this transcriptomic observation corresponded with other cellular phenotypes. If you go to the final slide. And the punchline of this is that it does, that these transcriptomically defined cell types have differentiable morphological and electrophysiological properties. So it's very predictive of both type, and also gradients within the types are also well predicted by the transcriptomics with other features. And that's these types that don't seem to be present in the mouse upper layers of the cortex are in fact highly distinctive. They're some of the largest cells in the cortex, and right neighboring to those are highly distinctive different types of cells. And so what seems to have happened is that as the cortex has expanded in area, the proportion and the properties and the diversity of cells that make connections across the cortex have similarly expanded. So I think this is really sort of a paradigm for what we can do here now, and for what we're likely to see, that there is this overall homology, but when you get down to the fine-grained levels of high granularity, you start to see many species differences that lead to functional specializations, which are important to understand in human in particular. Thank you.
Well, that was a terrific introduction, Ed. As you've all heard, Ed Lean has introduced human and non-human primate studies, and our lab has really been contributing to the development of the human cortex. The first slide is just a schematic showing that in the developing rodent cortex, for example, in the mouse, there are relatively few progenitor cells during the course of neurogenesis. And the next slide highlights the additional complexity and the prolonged developmental timeline of human cortical development. And probing the identity of these diverse cell types has really taken an enormous leap forward with single cell profiling and multi-omics. Next slide. So we've been studying the development of cell types from a molecular perspective, and more recently have focused on earlier stages, namely when the neural tube closes and neuroepithelial cells transform into radioglia, as shown on the right, and begin to generate neurons. Next slide. So we've profiled cells at a series of developmental stages shown in the left-hand panel, going from gestational week 5 all the way through to postnatal stages of adolescence, using whole cell sequencing in the earlier stages and nuclear sequencing for those later stages. And from these data sets, we've been able to identify a whole range of diverse cell types. And I'll give you just a few examples as we go through the next few slides. Next. One more, thank you. So at that earliest transition between neuroepithelial cells and radioglia, which the next slide will emphasize. Thank you, one more, I think. There, thank you. The transition from neuroepithelial to radioglial cells is accompanied by some very dynamic gene expression changes. If you look at, for example, the gene Notch1, which is shown in the blue line on the left, it's highly expressed in neuroepithelial cells, but then it essentially turns off when those neuroepithelial cells transform into radioglia. And that's validated in the panel on the right. If you look at the Notch1 column, at early stages at the top, there's no expression of Notch1. It then increases in expression at Carnegie stage 14 and 16, and then diminishes at Carnegie stage 22. So these dynamic changes in gene expression give us insights into what activated gene networks are involved in these different cell types and their transition from one form to another. Next slide. In addition, these early data sets have confirmed and expanded our understanding of the patterns of neurogenesis that occurs early on. So the middle panel, for example, emphasizes direct neurogenesis from radioglial cells into neurons, as well as an indirect neurogenic pathway that develops slightly later, by which the radioglial cells produce intermediate progenitor cells that then produce the neurons. And these patterns of neurogenesis are dynamic, and they can be measured and realized by studying these individual cells' transcriptomes. Next slide. A surprise was the appearance of many of the genes that are associated with neurodevelopmental disorders, and some of them are listed here, that actually appear at this very early stage of neuroepithelial cell to radioglial cell transition. And these neurodevelopmental disorders manifest usually after birth, but I think it's striking that they may have etiologies that begin at very early stages in brain development. Next slide. And one of the other features of this analysis is that we now have gene signatures that identify cell types. And the movie that you're seeing looped on the right-hand side is of a particular cell that's highly enriched in the developing human cortex that we call an outer radioglial cell. And one of the features that distinguishes this cell is its mitotic behavior, shown here, where the cell rapidly translocates and divides, sort of jumps and divides just during mitosis. And this is a characteristic behavior of this particular cell type. Next slide. And our single cell data has now allowed us to identify gene networks that are enriched in outer radioglial cells during development, some of which are listed here. And that allowed us, next slide, to discover that the same gene expression patterns have been described in glioblastoma, very aggressive adult brain tumors. And the next slide shows a heat map of genes expressed in aggressive glioblastoma surgical samples. And the next slide highlights in red these gene signatures for these outer radioglial cells I've just described, suggesting that there could be outer radioglial-like cells in these tumors. And so to confirm that, we received some fresh tissue, which we imaged. Next slide. And if you watch this image, you can see cells in this tumor actually jump and divide just like those outer radioglial cells in the developing brain. And this has allowed us to identify an enriched gene called PTPRZ1, which is in the pathway that is involved in that jump and divide behavior. And it's uniquely expressed in these aggressive tumors and could be a target for a therapy that could either treat the tumor or at least restrict its spread. Next slide. So we've continued our studies of cortical arealization through the atlas that we've been constructing of developing cell types. And we've looked at multiple brain regions, but I want to focus on the cortex, which was the subject of the companion paper in that recent Nature issue. Next slide. I'm sorry, next. Yes, thank you. So we've been able to cluster cell types across cortical areas during development. And those are the sources of data that I'm going to summarize at the end now in the next couple of slides. Next. One more. One of our findings was that gene expression across different areas of the cortex over time is not fixed, but actually changes very dramatically over time. So going from left to right are the periods of early neurogenesis, middle neurogenesis, and late neurogenesis on the right. And across from top to bottom are different brain regions. And what these lines are showing is that there's a shuffling of gene identity in the same cell type over time, and that the identity of individual cells is not fixed in their transcriptomes, but the identity actually changes, and it changes within individual brain regions. Next slide. Yeah, the next, this just emphasizes that the transcriptional regional identity is very dynamic. Next slide. In order to confirm those bioinformatic analyses, we've gone to in situ mapping of our genes with spatial transcriptomics shown here. Next slide. And next slide. And these confirm some of those gene expression differences that are areal specific, that is in different brain regions shown here in prefrontal cortex on the left, all the way to visual cortex on the right. Next slide. And a different way of conceptualizing our data is shown in these constellation plots. And if you look at the different cell types within a lineage, starting with radioglial cells, shown here in brown, they have a lot more in common with each other than they do with any aerial identity. And the aerial identity is written in lettering on top of these dots. So the radioglial cells don't really show very much regional differentiation. But their daughter cells, shown in brown, which are the intermediate progenitors, already have regional identity that's very distinct and different from the radioglia that produce them. And they're more related to the neurons that they produce, and particularly in V1 and PFC, that is visual cortex and prefrontal cortex. The gene identity of these intermediate progenitors already starts to match the regional identity of the neurons. And the neurons themselves have the most distinct identity from each other based on their location within the brain. Next slide. And that's summarized here, where on the left, at the lower cross-sectional diagram in red and blue, we've indicated that the prefrontal cortex on the left and the visual cortex V1 on the right, the two opposite poles of the developing brain, are very distinct in their gene expression differences, even at the level of radioglia, the progenitor cells. But with time, going here to the right, while those two poles remain distinct and very stable in their gene aerial identity, the areas in between, which include the parietal areas and the temporal lobe, those start to become more distinct aerially, not in the progenitor zone, but in the neurons that are produced. And so what we find is that there's a protomap in the progenitor cells, shown on the left, at the poles of the cortex, the front and the back, essentially, but in between areas emerge not in the progenitor zone, but rather in the neurons that they produce in what really is a proto-cortex hypothesis rather than a protomap. So this is evidence for a little bit of both of those processes and both of those hypotheses accounting for aerialization. And the reason that aerialization is important here is that the brain, unlike any other organ in the body, is very dependent on location for function. So the same neuron in one part of the brain will have a different function than that neuron in another part of the brain. And that's why we're very interested in seeing in these early atlases exactly how that aerialization begins to emerge. Thank you for your attention.
Great. So you've heard about some extraordinarily deep and diverse data, and that's motivated a lot of analysis that's tried to be correspondingly customized and thoughtful about the way to interrogate the data. Next slide. And so the central focus in a lot of these analyses has been a question that comes up in many talks, which is the number of cell types. And that's something you've heard about because for each data set, we have to think about, is variability across the data set driving novel cell types, or are we seeing the same cell type? And that variability can be biological. So across species, we have to think about whether we're seeing the same cell type. Or it could be technical, and often those two things can be slightly confounded so that it's hard to sample identically across species. There are technical covariates that will change as well. So the BRAIN Initiative obviously has enormous utility from a neuroscience-centered view in collecting coordinate data that is complementary and covers different modalities. But from an analysis point of view, it's potentially even more valuable, just because if you think about it, the major confounds we think about in analysis are experimental confounds. The central question always in science is, does this experiment replicate? And the central technical problem that one could be worried about is, does it replicate across laboratories? And so really no individual laboratory can conduct that analysis fully. It requires having multiple laboratories doing complementary experiments in their own best known modality or with their own best known technology, and then making comparative assessments of what holds up and what might hold up less so. And that really lets us distinguish between technical variability and biological variability. So the BICCN's coordinated efforts really provided a unique opportunity to probe the robustness of cell types and thereby establish biologically meaningful coordination across modalities or across species. And I hope actually that the data, just as the reference genome it has enormous utility, of course, in itself, but is really useful for methods developers to build additional analyses on top of it. I think the BICCN offers that same opportunity for the future. Next slide. So there were many analyses focused in various ways on integrating and comparative assessment across the different data generated. I've highlighted that this can take slightly different forms from one laboratory to the next. So one that many people are familiar with is cross-modality integration. So that comes up quite frequently within laboratories, where you have a batch effect, or it could be an experiment effect that you want to integrate across, and you have also potentially some biological variability you don't want to fit away. And then there's a second set of analyses that were done by a number of labs, which are particularly well powered within the BRAIN initiative, which is replicability and assessment of pipeline differences on called cell types. And then these first two are slightly more technically oriented. So they're trying to establish methods that work strongly, what features can then be associated with variable cell types or consistent cell types. So if you see the same cell type, does the feature set vary? If you see different cell types, are there any correspondences between them? And then there's strictly biological investigation, so identification of shared cell types across species, or continuous or discrete contributions to cell type identity and developmental. Next slide. So one of the results I wanted to highlight, because I do think it comes up quite often, is the word cell type is a word that allows for a lot of differing weight to be put on it. And it's easy to use the word kind of variably and to ignore the differences contained within it. And so there were a lot of analyses in particularly the mouse MOP paper by all the groups trying to capture replicability of the cell types. And so I actually showed three different ones here that all drew almost identical conclusions about the most robust cell types. And so one of the nice things about this is that it's highlighting that there really are quantitative scores associated with almost all the called information. And so there are subtler things that replicate slightly less well, and there are very, very robust things that replicate perfectly across all data sets. And that's useful if you're looking up markers for particular cell types, that sometimes what's most precise, meaning it's highly, highly specific to the cell type, isn't necessarily the most robust, meaning it actually fails in some experiments precisely because it's a bit fragile in how it's expressed. So there were a series of analyses, and again, in every paper, there's fairly detailed documentation of how these were done, establishing the replicability of cell types, and there are a particular set with marker genes associated with them that have a very strong degree of robustness and can be applied in other data sets with almost, I think, very strong confidence that it will generalize to new data. And I think finally, the point I wanted to raise is that this data is available, which you'll hear about in the next talk too, but even the raw data admits to a very, very detailed and thoughtful interrogation for new principles for experimental design. And I think that has a lot of value, and I hope other groups will continue to use this data for that purpose. And we're certainly happy to help if anyone reaches out. Thank you.
Thank you. Next slide. I'm going to tell you a little bit about our data access. The BICCN is generating vast amounts of data, and this can be impactful, not just by the analysis by the consortium itself, a lot of the exciting research you've heard presented here today, but also by continuing use and reuse in the future by other researchers who are asking different scientific questions, but can still tap into these data sets to really make great advances. In support of that, we aspire to making the data available and useful to others. So part of this is in the consortium is by our policies. We've had all of our consortium members have rapid release of their raw data to the data archives with no embargo. We require that our data researchers are providing protocols for the data generation, and we got some great support from that, from Anita Bandrowski at UCSD, helping us to orchestrate and educate people on how to use protocols.io. We also have all of the data is available under a CC BY 4.0 license. So this is a very open licensing that requires attribution, but is otherwise fairly open to support sharing and reuse. Part of this is by best practices. So we are aspiring to getting open access human tissue, although that's not always possible for all experiments. And then we have guidelines for how our researchers are putting out their data availability statements. And we gave a lot of support to our researchers during this publishing phase on the cell census of the primary motor cortex to make sure that we had adequate citations for the data. We've also been ensuring that we issue RIDs for BICCN resources and tools, again, through support from our colleagues at UCSD. These are research resource identifiers that allow people to give a unique identifier to the resource or tool they are creating, and then cite these in papers so that you can track citations later. So as part of this, in addition to the data that you've been hearing about, there's a lot of these non-data resources. So you've got the 97 published protocols and 26 BICCN resources and tools. Next slide, please. Because the data exists at various data archives, the Brain Cell Data Center at the Allen Institute created a data catalog that gives you a chance to have a filtered search for data collections of interest. You can filter by like a subprogram or grant, species, technique, etc., and get your list of projects and open for any one of those projects a project summary page that provides description, contributors, publishing information, and the lists of data collections that are part of that project with links to data archive access and other links as provided. This catalog can be accessed through the data tab of the BICCN portal and now includes a lot of great data. So we've got 16 species worth of data. It's largely human, mouse, marmoset, but we are starting to get a lot of other non-human primate data in there, as well as other mammals. 117 data collections, 30 million cells profiled by various techniques, largely transcriptomic data, but there's also morphological reconstructions. Over 2,000 brains have been profiled in terms of like whole brain imaging and other histological brain section sets, MRIs, et cetera. So it's really a great resource. It's a chance to come in, find your data collections of interest, and be able to go find that data at the different spaces. So next slide, please. Thank you. So links from these project summary pages take you to the data collection at our data archives. So our BICCN ecosystem is a distributed ecosystem. There's that BCDC central organizing center with the data catalog, and then all of our great partners at these data archives. So we have currently, each of these data archives has data storage capabilities and compute capabilities. You can also search for your data and access your data at each of these archives coming in there independently. And these three official archives in the consortium are NEMO, Neuroscience Multi-Omic Archive, PI Owen White, which offers sequencing data from transcriptomics, epigenomics, and whole genome sequencing. The NEMO archive also has integration with omics data processing pipelines hosted by the Broad Institute's Data Sciences platform. And they're also affiliated with another effort, NEMO Analytics, to provide visualization tools for omics data. The BIL Brain Image Library, PI Alex Ropolewski, takes imaging data and its derivatives, including various types of microscopy, 3D brain imaging, spatial transcriptomics, as well as morphology. And then there is the DANDY Archive, distributed archives for neurophysiology data integration. PIs are Ghosh and Halchenko, and they collect neurophysiology data like electrophysiology, some optical physiology, but they also aggregate data like MRI with closely interconnected histology and or cell resolution data. Next slide, please. Finally, in order to support this large effort on the mini atlas of the primary motor cortex, we created an additional splash page on the BICCN portal. You can find it under the Science tab. And this is where you can access the list of papers that were part of this effort, links out to their Nature papers, as well as you can see where you can go actually explore a lot of this data. As part of this large effort, which involved so many different members of the consortium, we had a lot of great partners, members of the consortium, as well as external parties that contributed to making this data viewable and visualizable on different platforms. So this has been a really rich data set. You can go visualize the data at various places such as the NEMO Analytics, CZI Cell by Gene, Raul Satia's Azimuth. We've got Catalyst, Brain Cell Methylation Viewer, UCSD Brainome, the Allen Institute's Transcriptomics Explorer, Brain Architecture Project, Single Cell Portal at the Broad, and neuromorpho.org that is also hosting some morphological reconstructions. So this is a great resource. I invite you to go check this out. And we do hope that the larger community is able to tap into this and find the data that is useful for their research. Thank you very much.
Thank you, Carol, and thanks to all speakers. That was just terrific. You know, this is a project that I think we're all very grateful to be interacting with each other and working with each other on. There's a long list of acknowledgements, which we can't possibly do complete justice to, but just a few things that we put down here, including all the laboratories and scientists and all institutions who contribute to all the hard work to this, who really did all the heavy lifting, to all the grant support, which I undoubtedly have missed some here from NIMH and NIH, the various U awards, the various R awards, et cetera, that really led to this. We also thank Program Officer Dr. Yang Yao, who has been intimately involved in the direction and management of the study from its inception. And again, I mentioned the speakers. I'd like to thank our communications team and the late Mr. Paul Allen for his vision, encouragement, and support, and all of you for your interest in attending today. So with that, I think we're going to move on to our panel discussion now. And I think Marianne Martone is going to help lead this.
Yep, I'm here. So can I ask all of the panelists to turn on their cameras, and I think you can stay muted until you're ready to speak. I will try to moderate that. Can I have the next slide? So I really want to encourage, we already have a couple of questions in the question and answer panel. If you're viewing by YouTube, just put it into the live chat and we're transferring them over. And I want you to feel free to ask questions on both the science that you've heard, the BICCN as an example, as we heard in the beginning, of team science in neuroscience, a new experiment in that. And also what the impact is going to be. So please put your questions in and I will try to get to as many of them as I can. I put some of these up and we talked to the panelists about this on some of the issues that I just raised. And I think we will touch upon, right, the idea of team science and have people reflect on this tremendous effort, challenges, lessons learned, also the impact of what it is that we've done, both in terms of human disease and our basic understanding of how the brain is put together. I think especially the cross-species work has shown some really very interesting similarities and differences. And then I think reflecting on how the neuroscience community as a whole can build upon what was done. But first, I want to get to some of the questions that have been asked by the audience. So we'll start with two simple ones, and then I want to get to the translational question. So the first question is from Mei-Jin Yu, and it is, how would this single cell spatial gene mapping relate to macro scale PET and MRI mapping? Who would like to take that one? Ed, go ahead.
Yeah, this is really a fantastic question, because I think it gets to the heart of, how can this single cell revolution relate to brain function and relate to fields that do non-invasive types of imaging in humans. And this is indeed quite a challenge, actually, I think, to bridge these scales. And typically the field has had somewhat of a schism between these two sides of looking at the brain. But I think that there are ways that these are starting to be brought together. And one of them is that, similar to how you heard about mapping to a common coordinate framework in a mouse, we also map the human data to a reference framework coming from the MRI field. So from an average template MRI. So all specimens get mapped to an approximate location, although they, of course, came from a dissected brain sample. So there's a limit on the accuracy. But as the human profiling begins to map across many parts of the brain, we'll start to get a map that can relate to the architecture seen by imaging types of studies. And I think actually this, in my opinion, this actually needs to be a driving goal of future human brain mapping studies, is that it really needs to be able to connect these two, because ultimately we're trying to understand the cellular basis of functional architecture. And to do that, we need to be able to map across these domains. So it's sort of in its nascent state now, but I think it will increasingly be a driving goal of these efforts.
If I might add, I certainly agree with what Ed said. And having information about cells, the molecular information, and therefore markers, it really does give us an opportunity to develop cell type specific PET ligands, which will then allow us to monitor cells, for example, those that might be more vulnerable in neurodegenerative diseases and the like. So I agree with Ed, this is really a great opportunity. We're at the very, very beginning with these foundational data sets to be able to link across not just these scales, but also the molecular information to use that information to identify what these cells are doing across different states, ages, disease in humans. But this is really, I think, a missing link that would really make a lot of these human imaging studies much more powerful than they already are.
You're muted.
Okay, yeah, go ahead, Hangui. Yeah. So I think this is a great segue into the next question that came in via YouTube. How does this info translate into regenerative medicine, such as repairing damage in stroke or cerebral palsy? What needs to happen to get there from where we are now, given this data? Arnold?
Yeah, perhaps I could begin. That's also a very interesting question, and one that actually has touched on some recent developments. Single cell sequencing is giving us the molecular fingerprints of individual cell types, and the ground truth of what these cells really, what their identity is based on molecularly. And this is information in the human brain that is already being applied to studies in regenerative medicine. For example, in Parkinson's disease, with cell replacement therapies that are focused on dopaminergic, certain classes of dopaminergic neurons, the identity of those cells is now much more specific than ever before. And people are looking at their stem cell derived dopaminergic neurons and comparing them to the ground truth of the profiles that people, for example, those involved in this BICCN effort have defined as those molecularly identified cell types. The same is true in the retina when people are trying to treat macular degeneration by creating retinal pigment epithelial cells in vitro in a laboratory and then actually graft them into the patients. Those efforts, initially at least, have not been terribly successful. And part of that appears to be that the cell types are not precise enough. And that information, again, comes from a comparison of those in vitro derived cell types to the actual in vivo cell types being profiled by studies like the ones you heard about today. So, at least that's one example of how the molecular data that's being generated in these studies can improve the regenerative medicine approach.
Xiaowei, do you have something? You were unmuted.
Sorry, I was unmuted, not for this question, but when I was looking at, when Ed and John was commenting on the PET and MRI, I was going to actually add a comment or a question about the scale, right? So indeed, mapping the cell type to the common coordinate framework where you also have MRI images and PET images, so that'd be very powerful. But the scale is also making things challenging because in the local region, there could be many different types of cells, right? And that still particularly makes the linking of cell type information to the functional MRI and PET difficult. One thing is perhaps thinking about how to improve the spatial resolution of functional MRI and PET more and more towards, cellular resolution so that things can be linked better. I don't know whether Ed has more insights on that front.
I don't have much to say about improving the resolution of functional imaging. I hope that things will go in the other direction too, of improving the scale that one can apply the spatially resolved genomics methods to be able to tackle those kinds of problems. And that actually might be one place where the non-human primate can be extremely informative for human, because if we think of a marmoset, a very small primate brain, it's something that maybe could be tackled with spatial transcriptomics methods of today.
No, I was more thinking about any local region could have many different types of cells, right? So that is why the scale connection between functional MRI and PET to the spatial information of the cell types could be represent a challenge. If a region that is seen in the functional MRI and PET to have a certain function or activity has only one type of cell, that's easier. But if it has a very highly heterogeneous environment with many different types of cell, that connection could then still nonetheless be challenging.
Not to drag this question for too long, I just want to add one quick comment about mapping cell types, spatial distribution cell types with functional imaging. I think another critical intermediate link would be the wiring diagrams, the connectivity patterns. I think that's also going to be essential and an intermediate step between cells, network, and then the whole brain function. Yeah.
Thank you. So back to our question about impact on disease, because I think these things are not unrelated, right? The challenges involved in trying to understand in humans what the cellular makeup of the brain is. But John, Ed, or Juju, do you have comments on this potential impact looking forward?
I'll defer to others if they want to speak, but I could certainly comment on it.
Go ahead.
I was going to defer to you, Ed.
Okay.
You got the data. Why don't you go first?
Okay, yeah. No, I really want to build on what Arnold is saying. I think one element of it is understanding the cell types, and that can lead to a way to try to, for example, induce cells to become a particular type of cell in a regenerative medicine type of strategy. But another thing that's come very clearly out of this molecular characterization, and in particular the epigenetic characterization, is it is possible now to identify short enhancers that are capable of driving cell type specific gene expression in the context of an AAV virus. And these AAV viruses are in common use in gene therapy now, but they're typically used in a very blunt way right now, with just targeting every cell for gene replacement. We now can identify these enhancers that are really drive selective expression. They're sufficient in of themselves to fit in an AAV and drive selective expression in particular kinds of cells of whatever transgene that you would like. And so this, I think, really leads quite directly to some potential clinical applications, where if you can identify the vulnerable cell type or the causal cell type for a particular disease, you can now treat that cell type as a target rather than a molecular pathway and deliver a therapeutic gene of some sort. And I think this is really possible now. Many of these enhancer regions that can be identified are functional in the same way across species, including human, using human tissues that we've been working with, for example. And so it's really plausible. And I think this is a little bit of a paradigm shift, perhaps, that this field will enable, is thinking of the cell as the target. And if we can identify the right cells, we can deliver new therapies to those cells.
Yeah, just to add what Ed just said, so for the enhancer testing and identification of the enhancers that are unique to cell types, I think it could be an iterative process. So right now we are using the epigenetic information to predict enhancers. And then we are actually carrying out high throughput analysis for verifying whether any of the enhancers could drive cell type specific gene expression. Now we can use that information again to feed back in our prediction model to better train the model to find other features to predict really enhancers with really specific cell type expression. So that's one thing I think is really exciting. And another thing I think with all the epigenomic information, we can start to really annotate using, for example, comparative genomics approach, all the SNPs identified in the GWAS study. So most of the SNPs actually fall into the non-coding region, and we know very little about their function and why they might be associated with certain disease. So applying those kind of comparative epigenomics approach, also just applying the epigenomic effort into the mouse, into the human samples, we can really start to annotate those non-coding SNPs.
Thank you. Anyone else? All right.
Yeah.
Go ahead, John.
I'll continue on these thoughts. These are great thoughts. One thing that I thought was really powerful from this set of studies was the comparative analysis across 3 mammalian species, and it really highlighted what's similar, but also what's different, and particularly what's different between the human and the other organisms that we as neuroscientists and scientists tend to look toward as model systems for disease. So it really allows us to, in a much more informed or principled way, really test our hypotheses more carefully and constrain them. If a cell type isn't in a mouse that you find in a human, then you may not want to consider that as a model system. That's one area. And what Ed and Zuzu are pointing to as well is the power of having this ground truth information that Arnold referred to, about having the molecular fingerprints of the cells and now functionalizing or using that information for additional inquiries. So in the case of gene expression and enhancer elements that are now being developed, this really does give us an opportunity not only to further interrogate these cells experimentally, but also to think about how we might be able to apply those in humans and ultimately in human gene and molecular therapies. And if I can make a shameless plug for the Brain Initiative right now, we have 3 large projects that we are in the process of standing up, some of which are really based very directly on the BICCN and the success of the BICCN, and they're interlinked. One is an extension of the brain cell atlasing project into big brains, into human brains. Another is developing whole mammalian brain connectivity maps. This is coming. And another project that has just started is the so-called armamentarium for cell type access tools, along the lines, not limited to, but along the lines of what Ed indicated, of creating more highly precise tools for targeting cell types in the brain using right now molecular probes, including AAVs that hopefully will be more precisely directed by these enhancer elements that have been identified through these molecular atlasing approaches. So together, the cell atlas and the connectivity map will provide the ground truth. The ability to access these cell types in a highly precise way will give us a way not only to further interrogate and test our hypotheses there, but actually to functionalize that information into, I think, ultimately gene and molecular therapy. So we're very excited about looking ahead toward these projects. And again, they're very much based on the success of this large team effort that was taken on by the BICCN.
Thank you. I think these next questions will go to Jesse and Carol. We have a question from Wang Zhu. What is the computational, or what are the computational challenges on this huge, complex, heterogeneous data collection? I think you touched on some of them.
Yeah, I guess just for fun, I'll invert the question. So it says, what are the computational challenges? And there are obviously massive computational challenges, but those exist in the biology. So the problem is that there are variable cell types, they are diverse, there are different ways we can measure their properties. It's going to happen that we have those measurements. And so eventually we're going to have to figure out what that all means. And so that was either going to happen as a field-wide exercise over the next 10 years as people collect disparate data and try to figure out how to integrate it. And that's a computational challenge that might be intractable. It might be tractable eventually, but it might take more than 10 years. Or there can be coordinated efforts. And so, well, the data is obviously very challenging in some really interesting ways. I'll mention what two of the prominent ones. I would think about this data as actually being much easier than the alternative. The alternative would be not to have parallel data across modalities or coordinated data. And then we have the exact same problem of figuring out what's going on, but it's much more unsolvable. And so I think the two major issues in the data probably are, classical ones going back to, Fisher's plant experiment analyses, which is confounding, that, you want to make comparative assessments across individuals, across species, across conditional variation of some type that's biological, and it's confounded to some degree quite strongly with technical variation. So if you have different genome qualities between two species, for example, or other properties like that. And so, there's a lot of effort that goes into resolving that. And again, requires you to do an awful lot of sampling to resolve that. And the second one is really defining a feature space that you can analyze the data with respect to in one dimension or another. So calling things as shared cell types, and so you see an awful lot of reference to using expression data as a useful reference for other modalities is very, very common, or defining cell types that are close enough. Again, how one defines that, is a matter of discussion, but that are close enough that one can then just think about the feature differences. The features ultimately define the cell types, so it's a statement that these are close enough that the feature differences are sparse. So those are, I think, ongoing things that people can continue to work on with this data and make progress in. But it's an unusually helpful data set, even though it's complex relative to previous data.
I could add just a slightly different kind of angle to this. Jesse's thinking a lot about that analysis, and then I'm thinking some of the other challenges we have are where we have this, as you said, as the question says, heterogeneous data collection. We have a lot of different types of data, and a lot of the requirement is that if people want to work together to analyze this data, it is agreement on those standards, agreement on what formats do we want this data to be in if we want to be able to do a joint analysis across things. And I think those are some of the things that our scientists and their working groups worked on in some places about how do we want to have this data in a similar format so that we can do some of these shared analysis. I'm aware of a lot more on the transcriptomic side, but I think a lot of that was happening on the image side as well. But some of our challenges also are that some of the raw data is really big. Some of this data is so big, you're not going to transmit it over the internet, right? And so I think in the future, some of our challenges is that people aren't necessarily going to download all the data to figure out what it is they want to analyze. And that's why a big push has been that the archives have compute capabilities next to storage, so that if we can get people to really come to this mindset of, okay, I don't have to download all this data to do it. I could start working somewhere else. I don't have to, I can analyze the data in the cloud rather than doing it all in my local servers. That could be a shift in how people deal with data for analysis, so that they can alleviate some of these challenges of having big data sets and you're trying to fish through and figure out what it is you want, and start reaching out to some of these archives or thinking about how you're going to deal with the stuff in the cloud so that you can run some of your analyses, filter through the data, figure out what's important to you, and get around some of these challenges. So I think that's a big thing that we're getting to in the future. I know in the future, there's going to be more of a push to having more of these pipelines for data processing that people can tap into. But it's moving the mentality of, I want to download data to, I want to use data, but I want it to be easier to use. So I think that's one of the challenges around doing this type of data project.
Thank you. Anybody else want to weigh in?
I again, not to take too much time, as I see that we actually have a lot of questions coming in. I just want to very quickly add one point in addition to what Jesse and Carol said. Not just the technical complexity of the amount of data and the diversity of the data itself. I think we also have to deal with the biological complexity. We see a lot of discrete, intermixing of discrete and continuous variations among the different properties of the cell types. And then in different modalities, in different ways of characterizing cell types, there are also very different scales of dimensions. How many genes are expressed versus how many morphological and connectional features can be extracted, how to quantify them, and how they are correlated with each other. So I'm really looking forward new computational methods that will allow us to resolve this kind of biological complexity.
Okay, thank you. And as you mentioned, we do have some, I think, really excellent questions coming through. So let's move on to one that I'm sure.
Can I say one other word on that? I'll just add in that just to link it to all the previous questions so the technical is not completely disjoint from the biological, that ultimately, we're going to have the computational techniques people develop to resolve these questions are going to be informed by the exact questions people were asking in the first, three or so questions. So that it's very important that people, try to use this data, try to answer targeted questions, and come back to computational researchers with these targeted questions, because ab initio, there just isn't an answer that is fully comprehensive that comes from on high. It really is resolved by having a targeted use case within the data, I think.
I think that's a very good point. So one question that I'm sure many people might have, and I had, is why did you aim at motor cortex, and what will be the next target? And I think, the question of generalizability of the armamentarium and approach to other brain regions are all really important questions. Who would like to take the first pass at that? I'm looking at Ed. He's shaking his head.
I think Hong-Kui would be probably the best to take this one.
Hong-Kui?
Okay, I'm happy to start to answer that. I think, why motor cortex? I think we would like to first focus on cortex because it's a very well studied structure. It's an intriguing structure that's greatly expanded during evolution, it harbors the higher order function of the brain. So first cortex, but then which cortical region? We chose motor cortex instead of other better studied regions such as somatosensory cortex or visual cortex, is that it's actually more consistent across species. It has it's more easily identifiable in human and non-human primate compared to some of the associational cortical areas. At the same time, it also does not have too much of a species-specific specializations. For example, somatosensory cortex in mouse has greatly expanded barrel cortex, whereas the visual cortex in human and non-human primate is much more specialized compared to mouse. So motor cortex is a much more better conserved, and we feel like there's probably a lot more common features across species. So that's related to what Ed is studying. And then the next question, what is next, right? So the next is actually we are making a giant leap now. We're covering not just another region or even multiple regions. Our goal is to cover the entire brain. And we're doing that in the mouse brain now. And we actually hope to get that done very quickly. So expect a lot of data coming across all the different regions of the mouse brain. And then, as John said earlier, there's already new funding opportunities available to allow us to move into characterizing the whole human brain as well, or other non-human primate brains. So a lot more data and information will be coming.
Thank you. Anybody else want to comment on that? If not, I think there's a follow-up really that says, how, given the rich functional data from the primary motor cortex during movements, how can we go about mapping this genomic information to function in motor cortices, such as during reaching tasks in mice? So again, mapping what we've learned to functional behaviors.
So I can take a first, answer at that. So, this, is where I mentioned that imaging approaches tend to be easier to reach the multimodality. So you could imagine that when you have these motor cortex functional data, or you can see that when it's during the reaching in a task in mice, then we can look at which neurons are activated, for example, by calcium imaging. And then afterwards process the brain and then do spatial transcriptomics approaches by, single cell spatial transcriptomics by imaging, and then map the cells and align them so that you can see what are the molecularly defined cells or the molecular features of the cells that showed the calcium activity data. So this way, then you can have a direct link of the functional role of the cells to their molecular features, or to link to the cell types to see which cell types are activated.
Thank you. So I think we're running out of time. I wish we had.
Could I just add one thing to that?
Go right ahead. Yeah.
I'm going to maybe channel one of our other consortium members who's not here, Josh Huang. Understanding the basic cell types then lets you ask the next question, as we heard about where do they form connections to be part of functional circuits, and also to generate tools to target them. And so that leads really directly to now a path to start to do interventional experimental neuroscience about the function of particular cell types in behaviors and in motor behaviors. And that's actually, I think, what Josh is doing a lot of now is starting to have selective genetic access that lets you manipulate different kinds of cells and understand what their functions are. And there are different functions of different cells within the cortex that's highly related to their connectivity. So I think there's really a nice path here from, again, the basic characterization to starting to understand the selective roles in behavior.
Okay, thank you. So we're down to our last 5 minutes, and this last question is, and I apologize for the questions we didn't get to, and I hope that the panelists will engage and answer some of those so that you have them. But this one really thanks you for this tremendous and beautiful collection of work and this efforts really to pool and share data. And there's a lot we didn't touch on, such as standardization of morphological features and others. We know that there are those challenges. But she's asking basically that there are so many misunderstandings of regional brain functionality in popular discussions. Cross modalities alone are not well communicated in public arenas. And the BICCN seems well poised to wonderfully explain aspects of scale and connectivity and clarify the progress of knowledge about circuits and regions in the mammalian brain. So how does this work really fit as a theoretical map for where we are now in the knowledge space? What do we know and what is left to know? And I think maybe we could just, you can unmute your microphones, or I can call on you just to give a very brief reflection on this, because we don't have much time. Who would like to go first? Arnold, what do you think we know and what do we not yet know?
Right. Well, I think
1 minute.
I just want to comment on one of the aspects of that question, which is a pretty detailed question, and that has to do with public outreach. I think it's really incumbent on all of us to communicate some of these findings in a very understandable way to the public. The public endorsed the Brain Initiative effort as a program for cures, and I think that at some level there needs to be a dialogue between us and the public in terms of how these efforts are achieving those goals. And there are lots of different ways that they are, but I think the public really has so far not been informed about the progress. And this flagship publication was a wonderful way to inform the science community, but I think it needs to be coupled to some sort of a public effort as well in the next few years, and especially as the Brain Initiative matures.
Thank you. Who else would like to comment? What do we know and what do we not know?
I think one thing it's really exciting for me is from this rich data set together, we know that the cell type are not really discrete definition, but really a continuous definition between there are quantitative difference between different cell types and for example, from different brain regions. But what we don't know is how they are established. So where do we have this quantitative difference as a gradients across the cell of mouse brain? So I think that could be one of the very exciting questions next, to understand how they are established during the development.
Anyone else?
Yeah, I'd like to continue on Arnold's thoughts. I mean, I definitely do strongly believe that this is our opportunity to take this great foundational data set and with many more to come, to functionalize it and really drive it toward cures. There are so many devastating neurological diseases out there and neuropsychiatric diseases for which we have no hope until now. And I really do think having this information together with these new molecular tools that are being developed, together with other revolutions going on in parallel, for example, in CRISPR gene editing and other modalities. I think we do have a chance to do this, and we do need to be clear to the public that we are working on this, and to be very clear about our successes and our commitment, most of all, toward these eventual cures. I actually think we can do things in our generation that I think 5 years ago were deemed to be essentially impossible. And with the great data, ground truth data, and foundational knowledge we have now, and that will be coming in the next few years, focused on human brains, I think the future is looking much brighter than it was before. But it's going to take a lot of hard work, a lot of talent from many, many people across many, many different fields. One of the really amazing things about the BICCN is we had people from all walks of life. We had a bunch of plant biologists, a bunch of biostatisticians, informaticians, in addition to what you would consider traditional neuroscientists, a physicist right here, right? So I think these are the kinds of collaborations that will really make the next 5-10 years particularly more exciting.
So thank you, John. And I think on that note, we'll end the panel discussion because we are out of time. But I want to thank the panelists, and I want to thank the audience for your very thoughtful and provocative questions. So now I'm going to turn this over to Mike, who's going to wrap this up.
Yes, thanks everybody for a great webinar, very informative for me. I learned some things. Don't hesitate to follow up with us. You can send mail on general questions to BICCN.org, web mailing list, right? Or you can write to individual investigators, should you like, or Marianne. We will be in touch with more progress in the near future. Well, thank you very much for attending.